Hi guys, welcome to my YouTube channel. If you are new here, subscribe to be notified as soon as I upload a new past paper video with simplified explanation. Don't forget to like and comment. I will be happy to solve any doubts. Today we will be discussing Cambridge IGCSE 0610 paper 6 variant 2 from October-November 2017 session. Now, to start with this, we are talking about the paper where the question reads as that starch is an important food source that is digested by the enzyme amylase to form reducing sugar maltose. Now, just to brief you up as to what exactly is happening over here. So we know that starch is a plant product or a storage form of plant. So here, this glucose is converted to starch and it is an important food source that is digested by the enzyme amylase, which is present in the human body, in the mouth, as well as in the uh, intestines. That is, it comes from the pancreas. Okay, so it is forming the reducing sugar maltose. Now, maltose, when further digested with the help of maltase in the human body, it will be converted to glucose. But here, the experiment is stating that it is starch plus water in the presence of amylase as an enzyme will form maltose. Some students investigated the effect of enzyme concentration on the rate of digestion of starch. Now, this is the point where we start identifying the independent and the dependent variables. So, we are talking about the rate of digestion. Now, this is something that you can measure, and what we can measure is what we are going to talk about as our dependent variable. The concentration of the enzyme will be our independent variable. Now, moving ahead with the question, it reads that step 1, take three test tubes which are labeled as test tube A, B, and C. 5 cm cube of starch solution is put in each of these test tubes A, B, and C. So we are going to just keep in mind that it is 5 cm cube. Another three test tubes are labeled as A1, B1, and C1. So it's A1, B1, and C1. 1 cm cube of 3% amylase solution was put in A1. So here we have got 3% amylase. Amylase, as we remember, is the enzyme to break down starch. In the second one, we have 2%, and the last one, that is C1, we have 1%. All six test tubes are placed into a water bath at 60 degrees Celsius for 3 minutes. So, we are going to remember these points that it is placed in a water bath at 60 degrees Celsius for 3 minutes. A white tile was divided into three sections, as you can see here on the uh, diagram provided, that it forms two rows of 10 drops approximately the same distance apart in each of these sections, that is A, B, and C. They have shown you that there are 10 rows which are present over here, and in each row the iodine sample was dropped onto the tile to form two rows of 10 drops approximately. Okay, so that's what we are saying, that here they have added the iodine solution. Okay. Step 8 reads as that the glass rod was dipped into the starch solution in test tube A. So we remember we had the sets of test tubes which were test tube A over here. Okay, and then we have this enzyme amylase which is present in A1. So The glass rod was dipped in the starch solution in test tube A to remove some of the solution. Remember that this is not measured, it's just a glass rod which is used. The glass rod was then touched on the surface. So, as we understand that the glass rod was touched on the surface like this, okay, and then a second drop of iodine solution in row 1 on the section of the tile labeled A. The glass rod was rinsed and dried, so this was a precaution that they had taken so that there is no contamination. Step 8 was repeated using the amylase solution in test tube A1 and the drops of iodine solution in row 2 on section of the tiled label. So here in the second section, along with the iodine solution, what they have done is they have also added the amylase solution. Okay, so when starch is reacting with amylase, that is it's broken down by amylase, okay, the iodine solution color will change. Initially, in the presence of starch, the iodine solution will show a positive result. So the positive result will be blue black color, but when the enzyme amylase is added, the blue black color will disappear. So, when amylase was added, the starch has been digested, and this digested starch has now become maltose, which is going to show a color change. So now iodine will only be showing blue black color in the presence of starch. If starch is not there, it returns to its original color as yellow black. Okay, now step 8 was repeated and the timer was started, and the amylase solution in test tube A1 was poured in the test tube A. Okay, so now what they are doing, a timer has been started and amylase solution in test tube A1 is put into the test tube A, which was containing the starch solution. The mixture of starch and amylase in the test tube was stirred with a glass rod, and then the sum of the mixture was immediately removed using a glass rod. The glass rod was then touched on the surface of the first drop, then the second drop in solution in row 3. Okay, so here in row 3 we are talking about is this is where they had added the mixture of the amylase and the starch solution. The glass rod was then touched on the surface of the first drop, then the second drop of iodine, and in the third row of the section of the tile labeled A, the glass rod was rinsed and dried. After 1 minute, the glass rod was used to remove some of the mixture. So after 1 minute, a glass rod was used to remove some of the mixture from test tube A and touched on to the first drop, then the second drop and the iodine of the iodine solution in row 4 of the section and the tiled label A. The glass rod was again rinsed and dried. Step 11 was repeated 6 more times. Step 8 to 12 were repeated for test tube B and B1 and C1 simultaneously. Now the figure is showing the results that the student recorded or observed. So they have provided you a key over here which says that there is starch or there is no starch. Okay, so where we can see the dotted lines, the starch are still present, and in the no dot, the starch has been digested. The first row, as we understood that the starch solution was added, second row, it was mixed with amylase where the um, starch was digested to maltose and hence the color was changed. Okay, now prepare a table to record the students' results. The table should include the concentration of amylase solution and the time taken for all the starch to be digested for each amylase concentration. Okay, Now, we always start the table by writing the independent variable on the left side of the table. So here, this is my independent variable. Whatever I observe is going to be my dependent variable. So as we go back to the question where we had already marked this, so we said here concentration of enzyme is the independent variable and the rate of digestion will be the dependent variable. So we had three concentrations. So here they had mentioned about this, that there was 3% amylase solution, 2% amylase solution, and 1% amylase solution. It is preferred that you write the format in the same way, that is 3, 2, and 1. Now the results are shown. So that is the time taken for the starch to be digested, which is recorded in minutes. Trial 1 and trial 2 was conducted. So here we are going to measure the timings that are seen. So on row A you can see 1 and 2. So that's how I have written it as 2. Okay. Then for the trial 2, again the result was same. For the 2% amylase solution, we can see that the reading was for 1, 2, 3, 4, 5, and a little bit is spilled over here. So 6 is what I have written. Okay, and then we are talking about the second one was 1, 2, 3, 4, and 5. So that's how I have written 5. And the third, that is 1% amylase solution. So it's 1, 2, 3, 4, 5, 6, 7. And the remaining here, there was one drop which was empty. So that's 1, 2, 3, 4, 5, and 6. Okay, so that's how 7 and 6, that is how the table is made. Your table is marked according to your independent variable, dependent variable, and all the six units or minutes that are measured. We move to the second part of this question, which is saying that explain why the students' results are not reliable. For here, the answers which could possibly be accepted was that the drops for B at 3, 4, and 5 have been merged. So we don't know whether it is 5, whether it is 6, etc. So it just raises a confusion. Result for C have different end times. Okay, so here it is showing that it's ending at this point. Here this point is empty. The result for C is different at 3 minutes, or there are no repeats, which is one of your safest answer when a question like this will appear. So we move to the next question. The question reads that the starch is digested into simple sugars. That's exactly what we started the question with, that the starch is a carbohydrate, which is a polysaccharide. When it is broken down by the enzyme amylase, it is broken down to maltose and then to glucose. So they are saying, describe how you could test the liquid in the test tube to show they contain reducing sugars. So we need to write the steps clearly. Take a 2 cm cube sample from each test tube. Add 2 cm cube or the same volume of Benedict's reagent. Benedict's reagent is the reagent that we use to find whether the sample contains reducing sugars. Heat it in a water bath at 80 to 95 degrees Celsius for 2 to 3 minutes. This is a very crucial step. Most of the time, the mark scheme will mark you only for these two steps, which is the naming of the reagent and the heating process, which is a heat in a water bath. Observe the color change from blue to brick red. So here for reference, I have shown you the image that the blue original color to brick red as the final color. Next question. State one variable that was kept constant in this investigation. So describe how the variable was also kept constant. So one mark is for identifying the variable and one mark as to correctly mentioning how it was kept constant. If the variable is correct and your how to do the procedure is incorrect, you will be marked 1 for the same. Okay, so let's understand there were many variables which were kept constant over here. The first thing we spoke about was the volume or the amount of starch solution, which was written in the question over here that it was 5 cm cube. So here it was mentioned that it was 5 cm cube. Then the concentration or the amount of the starch solution could be another option. So same concentration of starch was used each time the test would be repeated. Then we could talk about the volume of the enzyme amylase. Again, you saw that all the three percentages which were there, 3, 2 and 1 were all kept at 1 cm cube. The temperature could be maintained at 60 degrees Celsius was also mentioned, and the time, that is 3 minutes for equilibration, okay, was also mentioned. So all these are your options. Explain why all the test tubes were left in the water bath for 3 minutes before amylase was added to the starch. Now, for any experimental procedure, this is a very important step, that you try to equilibrate the temperature for all the um, test tubes, or else it may show a variable result. So your answer will be that all the test tubes reach the same starting temperature, or they have the same starting temperature. Okay, now moving ahead, explain why step 9 was carried out before mixing the amylase and the starch together. Now, step 9 was when they had placed only the amylase over there. So, to confirm that there was no starch in the amylase solution, if this is a kind of a test to prove that your experiment is valid, and also to check for any type of contamination in your thing. If amylase if starch was present in the amylase, the color would have been different. Okay. Now, identify two sources of error in step 10, 11 and 12. Now, there are again many errors, but it's a 2 marks question. So what I have done is written 2 points, but I will tell you all the other points. So judging the eye of the judging the color of the endpoint by eye. Now, this can have a problem because it is a judgmental procedure. So, what we could do is that, we could have used a standard for, like, a colorimeter or like a chart for comparison. Another error could be that the timing and sampling, everything was happening at the same time. So, what we could do is that start the timer, then mix the sample, and then note the time when the sample was taken. So that will help you to reduce the errors. Another thing was that you are using one drop of both spots of iodine. Okay, so you could use two different rods, or you could also use a pipette so that the size of the drop would not cause a change in the final result, because the drop size is variable. There could be contamination, so that is why, to avoid contamination, we could use two rods. Two samples were taken at the same time, so you can use two glass rods again, or take two separate trials. As I mentioned earlier, that the drop size of the iodine from the was taken from the glass rod, so we could use a pipette or a syringe for making it a standard or a proper reliable procedure. So I have given you the this question asks you that, for one of the errors you've identified, describe how this method could be improved to reduce the error. I have given you a list of all the errors that we could speak about. Next question. In another experiment, some students made starch agar that contained 100 milligrams per cm cube of starch. The starch agar was stained using iodine and then cut into blocks that measure 2 by 3 by 0.5 centimeters. Calculate the total mass of starch in each of the blocks of starch agar and show your working. Now, since it is a rectangular block, we will be using the volume of the rectangular, which is length into breadth into height. We have to substitute the writings over here. So the volume will be 2 into 3 into 0.5, which is 3 cm cube. Now we are talking about the concentration into volume because we want to find the mass. So 100 into 3 will give you 300, which is 300 milligrams is the final mass that we are expecting to work with. 6 small beakers containing 20 cm cube of 5% amylase solution was placed in water bath at different temperatures. One of the blocks containing starch from D1 was placed into each of the beaker. So what was D1? So D1 was your, okay. This is a very important point to remember, or read the question thoroughly. If you do not read the D1, you will have no idea where the further calculation is coming from. Was placed in the beaker and the time taken for each of the stars to disappear was measured. So the temperature kept on increasing from 20 to 70 degrees Celsius. Time kept on decreasing, which was measured in seconds from 1500 to 88. The rate of the reaction per milligram per second is what they are measuring. So complete the table 1.1 by measuring the rate of the reaction at 70 degrees Celsius. Now, from D1 we know that the size of the cube was 300 milligrams. So your simple working over here was 300 divided by 88, which is the time taken for starch to disappear. When you divide this, your answer will be 3.4. Write your answer in the table over here. Do not leave this empty because the table was looking something like this. If you have written your answer, if you have not written your answer over here, you will not get marks. Okay. Also follow the trend of the table because it is one decimal place. So your answer will be 3.4. Next question reads out that plot a graph on the grid to show the effect of temperature on the rate of reaction. We have to identify the x and the y axis because in biology papers this is not given to you. Always remember the left side of the table, which is your independent variable, is always your x axis, and the right side of the table or the measurement which you are observing is your dependent variable, which is always your y axis. Okay, so we have to plot the graph. The rule of plotting over here is that you have to use at least 50 or above 50% of the place provided. So you have to plan your graph accordingly, the scale accordingly, and then your marks are allotted. 1 mark is allotted for the axis. Okay, so we have identified and labeled it properly as your x-axis, which is temperature, and your y-axis, which is the rate of reaction. Don't forget to write milligrams per second. As I told you that the second mark is allotted for the scale. So your scale, you have to use at least 60 to 70% of the scale. Okay, the third mark is given for the plotting. So all the given points are plotted accurately. Okay, and then the last one is the line. So the fourth mark is for the line, which is the line drawn with a ruler or point to point, because they have not mentioned whether it is a line graph. Even a smooth curve is allowed. This will fetch you 4 marks for the graph. Moving ahead in the question, figure 2.1 is a photograph, micrograph of an epidermis of a leaf. It shows epidermal cells, guard cells, and the stomata. If we remember, epidermal cell is the outermost part. Guard cells are the ones which will control the opening and the closing of the stomata. They are bean-shaped structures, and stomata is the gap which is present over here in between two guard cells. Each stoma is surrounded by two guard cells. So we have 1 and 2, again 1 and 2. So they are surrounded by two guard cells which are containing chloroplast. You can see the chloroplast over here in the diagram. Now, epidermal cells are labeled chloroplast, cell wall, and the guard cells. Complete the table 2.1 to show two visible differences between epidermal cells and the guard cells. So first they have asked you to mention the feature. So we have spoken about the shape which the epidermal cells, you can see they are like very precisely shown as they are wavy lines, and the shape of the guard cell, as I mentioned, is a bean shaped structure. Okay, so this is what we are looking for. The next, obviously, it is shown that the chloroplast is present in the guard cell, and as we know that the epidermal cells are transparent, so we can say that the chloroplast is absent and present. More points that you could talk about was the cell wall, that there is thin cell wall here in the epidermal cells, but it is very dark, and that is how we understand that it is a thick cell wall on the guard cell. The size of the cell, the epidermal cells, they are quite large, but the guard cells are smaller because they are all like magnified to the same scale, so we could understand that as well. Okay, and the cell arrangement, we could say that the epidermal cells are not paired, but the guard cells are paired. So all these would be your valid answers. Now next is your drawing, which is a compulsory kind of a question for your biology ATP paper. Make a large drawing of two guard cells and the stoma shown inside the box in figure 2.1. So you have to see that there is a one mark for single continuous line. There is no shading allowed in this diagram. They have mentioned very precisely that they want to see only two guard cells and the stoma between that. The drawing should occupy, so pay attention when we understand this, that this is your line or the part which has been provided. You have to follow the margins according to what I'm drawing. Okay, in between, your drawing should occupy approximately 70% of the place. Don't change the orientation of the drawing as well. Okay, so one mark is for your smooth outline, then the space occupied, so it should occupy at least 70% of the place. So in your paper, if you take a printout, it should be more than 55 millimeters. Okay, then we are talking about the stoma. Width is about 1/6 of the width of x and y. So what is x and y over here? So here your x and y is the out to out measurement of this stoma. So if this is what I measure from year to year as 50 millimeters, okay, then it should be at least 1/6 of the available space, and the cell walls should be drawn. Okay, so because it is a plant cell, and we can see that this should also be proportionate, that this should be thinner as compared to the inner ones. These are the thicker cell walls. Okay, so all these details need to be focused when you are marking, when they are marking you for the 4 marks drawing. Now, measure the total width of the guard cell and the stoma along the line XY. When I took a printout, I was getting around 32 millimeters. That's the total width. Now, they have asked you to draw a line on your drawing at the same position as the line XY. So, since this is mentioned, we have to draw the line XY, which we just spoke about. You have to label it here again as X and Y. Okay, and then measure the width of the guard cells and the stoma in your drawing. Always remember, for the measurements, you will always use millimeters for this particular kind of questions. So millimeter is what we are going to write. So total width of the guard cell and stoma in your drawing is 58 millimeters. We have been provided the formula for magnification, which is width of the drawing upon the width of 2.1. So we just have to substitute our units, that is 58 upon 32, which is approximately 1.8. But they have specifically told you to write the answer in the nearest whole number. Don't forget to read this out. Okay. Now, when we are writing magnification, we means that it has been multiplied or magnified this many times. So you always have to write an x and your final answer. X means it is multiplied or magnified that many times. Okay, so that's how the answer is got. Now the next question is, figure 2.2 shows the rate of water gain by absorption and the rate of water loss by transpiration in a plant during a 24-hour period on a hot sunny day. Now, as we understand that the water gain will be done by the roots by the process of osmosis, we have the root hair cell which is adapted for its function of absorption of water. It has a large surface area for absorption of water, and water travels down the water potential gradient from the soil into the root hair cell. Okay, so and the loss of water by transpiration. So if we remember, transpiration is the loss of water in the form of water vapor through the stomata. This happens through in the daytime when the plant is photosynthesizing or when the stomata is open during the day. How is the stomata open? So we remember that the stomata is having these two guard cells. So when the guard cells become turgid, they will uh, op uh, open the stomata because the inner walls are thicker as compared to the outer walls. When the guard cells are flaccid, the stomata closes. Okay, so this is how we understand that the guard cells are responsible for the closing and opening of the stomata. Now the graph is reading a rate of absorption. So here they have shown you the dotted line as absorption and the other as transpiration. Okay, now compare the trends shown in figure 2.2 for the absorption and transpiration of water in a 24-hour period. Now whenever a question like this is provided, it is important for you or it is mandatory for you to use the data from the graph provided. So try and start your answer with a conclusion stating that absorption rate is lower than transpiration at 9 to 18 hours during the day. Transpiration rate increases faster than the absorption rate as the day moves from 10 to 12 or as the heat intensity increases. Absorption peaks at 18 hours. Don't change the format if it's written 1800 hours or 18.00 or it is written 6 o'clock. Whatever, if it's written in this format, you write this format. If it is written in the .00 format, you follow that format. Okay, so we are saying that the it has reached to a peak at 18.00 and transpiration peak between 14 to 16. Okay, so you can write these units over there. Moving to the last question, this is a 6 marks question. So figure 2.3 is showing the apparatus used to measure the water uptake by a leafy shoot. The leafy shoot is tightly sealed in a glass tube which is connected into a capillary tube containing water. As the leafy shoot loses water through its leaves, it absorbs water from the apparatus. Air is pulled into the open end of the capillary tube as the water moves towards the leafy shoot. So you will see that the water will move ahead. The distance moved by the air in the capillary tube, which will be your dependent variable, can be measured on the scale and is used to calculate the volume of water absorbed by the leafy shoot. So let's understand this apparatus first. This apparatus is called as a potometer. This experiment belongs to the transport in plants chapter. So here we are talking about when transpiration takes place, the water vapor will be lost from the leaves. In turn, the leaf will take the water from the xylem in the stem, which will try to take it from the roots. But since there is no root over here, the xylem will just keep on continuing the absorption work. It is a cut end over here. Here you have put a rubber cork to assure that there is no water loss in this apparatus apart from the leaf. There is this capillary tube, okay, which is having this water filled, as they mentioned that as the water will be absorbed in this part, what will happen that the air will enter into this tube and it will move the water ahead. So that's how we will see that at the start the water will was here. After some time, I assume the water will be here, and that is the measurement that you're supposed to take, the distance between the two. Okay, now here that you can see, this is an apparatus which is attached to the potometer, or it is a part of the potometer where the water is filled. There is a cork which is provided over here. The main function of this is to refill the capillary tube so that the, okay, I will show you. Okay. So when we want to repeat this experiment, we will have to fill the water back up till here. So we open this cork from here, and then we reposition the level of water. So it's to refill the capillary tube or to reposition the water level to the start. Okay, so that's the work of this particular tube, and to reposition the water level. Now, the question states that describe how you would use the apparatus in figure 2.3 to investigate the effect of temperature. So we are investigating the effect of temperature, which is our independent variable, or humidity on the rate of water vapor by the leafy shoot. So we start the experiment always by writing the independent variable. There are three different temperatures that we are going to measure. I am sticking to the temperature, so that's how we are going ahead. Dependent variable will be the measuring the distance moved along the capillary tube. Now the experiment will be set up as similar. So arrange an identical setup of potometer to keep the apparatus in three different rooms with a temperature which is maintained at 50 degrees, 30 degrees and 45 degrees Celsius. Use the same type of plant with the same, okay. So here the same type of plant, the same number of leaves, the same type of light intensity. All this will be your controlled variables. You are supposed to write at least three controlled variables in your experiment. Okay, so here we spoke about the type of the plant, the number of the leaves, and the same type of light intensity. Let the plant equilibrate for 5 minutes and then measure the distance of the bubble in the capillary tube at fixed periods like 2 minutes for 10 minutes. Always mention everything. Don't say that measure it for some time. This is not the way we perform an experiment. We need exact numbers. Always write the repeat procedure. So repeat the experiment 3 times. Reposition the bubble to start every time. Now, whenever we write this 6 marks experiment, you have always come across something which is called as the safety precautions. But since here we are not using anything like, a hot apparatus and acid, etc., there will be no mark for the safety precautions. So then your point that you could mention over here was to reposition the bubble to the start every time we are repeating the experiment. I hope you have understood this experiment, and if there are any doubts, do leave comments and I will be more than happy to solve your doubts. Don't forget to subscribe this channel and wait for some other videos which I will put up. Also give me suggestions on which specific papers you want me to solve so that I can go ahead. Don't forget to subscribe and like the video. Thank you.